Hi, hello, good morning everyone. I'm Dr. Harika Kishore. On behalf of Vista's Learning, I'm here as your biology educator. Learning, it's an online education platform which works with an ideology of providing quality education for all. Visit our website www.v-learning.in where you're going to get an access for all the live classes, recorded sessions and also study materials. Moreover, you're going to get assessments too. And also, you're going to get this lecture notes in the form of PDFs. Get your acquaintance with all the channels made available for you and subscribe to the channel Vista's Learning Class 11 and 12. If you find this session useful and interesting, make sure you click on the like as well as share this session with your friends. And also click on the bell icon so that you are going to get all the upcoming video notifications. Saying that, let's begin at today's session. So as we already know, we have been in the series of the sessions where we are discussing about the chapter that is molecular basis of inheritance, which comes under the unit genetics and evolution, right? Now, so let's move forward where you have to discuss about this molecular basis of inheritance. In that, we already discussed about genetic code. Now, let's relate with the mutations. Here is the topic, that is mutations and genetic code. Mutations is nothing but a phenomenon where there is a variation in DNA. That is, there is a change in the sequence of the DNA which shows its effect on the genotype as well as on the phenotype of the organism. So, the relationship between genes and DNA. Genes are nothing but the segment of DNA or best understood by mutation study. That is a sudden changes that is going to happen in the DNA sequence, what we call it as a DNA variation. It may be caused due to so many reasons like exposing to the chemicals, UV light. So all this sometimes results in some deviation, some variation, and that sudden changes what we consider as a phenomenon of mutations, which results in change of the genotype, thereby changing the phenotype, that is the external appearance of an organism. So here we are here to understand the relationship between the genes and DNA, right? So here is a gene segment of DNA and we find here DNA, right? So here effect of large deletions are in insertions are sometimes and rearrangements in a segment of DNA are easy to understand, easy to comprehend. It may result in loss or gain of gene, which finally results in a loss of function or gain of function. That is what unso a function. As we already know, see, DNA which stores the genetic information from one of its strand which act as a template strand, we are getting mRNA. That means whatever the information that is stored in the DNA now been transformed into mRNA. And finally, it translates into proteins. So, whatever the change that is happening at even a one single base that is present in the DNA shows that change is going to carry forward. Therefore, there is a change that is going to appear in the bases that are present in the mRNA too. Here, if there is a single base pair that is changing, that is mutation going to happen in the DNA, It is going to carry out because it is the, that is a DNA is a genetic material where we can see the storage of the genetic information. So the same change is going to carry forward to the mRNA. Now that change will show the change in the amino acid, which is going to be present in the sequence of the amino acid in the structure of protein. So whenever there is a change that is going to happen in the gene level, it is going to reflect at the mRNA, and finally it is going to reflect in the structure of the protein. That is what here it may result in loss or gain of gene and so a function. Finally, the protein which is produced, which is translated, the mRNA which is further going to translate into protein, and that protein will show biochemical activity, biochemical response, right? Response, that means biochemical function. That is what they mention as. So as the final function is going to show the effect if there is a change at the DNA, that is the genes. If there is a change in the gene level, it is going to seen at the level of the mRNA, it is going to reflect at the level of the protein. Now, so here, to understand this better, the effect of point mutations, there are so many mutations like frame shift mutation and even point mutation. What base is replacing which base? Depending upon that, this point mutations are also of different types like transversion mutation and transition mutation. So here, to understand this point mutation, here is a classic example, and that classic example is sickle cell anemia. So here, point mutation where a single base pair change is going to happen at the DNA level. So a classic example of point mutation is a change of single, that is what we consider it as a point mutation. So single base pair change. A single base pair changed at the level of DNA, and the same change is forwarded at the level of mRNA 2, and it is going to change amino acid. The single base pair in the gene for beta globulin chain of hemoglobin that result in the change of amino acid residue. So change in single base will be reflected by a change in amino acid residue. So here, sickle cell anemia is due to a single base, nitrogen base change happened in the DNA that reflects in the mRNA, and finally there is a single amino acid change happened in the beta chain of hemoglobin. So here, if you once recover the structure, remember the structure of the hemoglobin, we already know hemoglobin is a tetrameric globulin, that is a protein. It's a pigment which is made up of 4 chains, out of which 2 are beta chain and 2 are alpha chains, right? So here, here is a protein part and here is a heme part. So we have 2 alpha globin chains and 2 beta globin chains. So here alpha 2 and beta 2, that is totally tetrameric structure we can see in the case of hemoglobin. So in the hemoglobin, there is a change that happens at the beta chain, not at the alpha. So the alpha chain will be remained the same as a normal hemoglobin, which is represented by HbA, A for adults. And the sickle cell anemia patients containing hemoglobin is represented by HbS, S for sickle. Now, a classic example of point mutation is a change of single base, right? That single base which is going to be happen in the gene, that it is going to reflect in the change in the amino acid that is present in the beta globin chain that results in the change of hemoglobin residue and that amino acid residue, and that amino acid which is going to be changed, that is from glutamate to valine. It is going to change from glutamate. It has to be glutamate in a normal hemoglobin, but it changes to valine, that is going to appear in a sickle hemoglobin. Now, so I already mentioned point mutation is of whether purine is replacing a purine or purine is replaced with a pyrimidine. It is of two types. Like one is transversion mutation, and one is transition. Transition happens if purine replaces purine. That is in the place of purine, again purine is coming. That means we know that what are the purines? Adenine, guanine. These are purines, right? If in the place of adenine, if guanine is coming, or in the place of guanine, if adenine is coming, here purine is replacing purine. That type of mutations, we call it as a transition. If in the case where purine place is occupied by the pyrimidine or pyrimidine place is occupied by the purine, then that type of point mutation, we consider it as a transversion, right? Now, let's come back to our classic example of point mutation, that is sickle cell anemia. By this time, we know that the structure of the hemoglobin is composed of 2 alpha globin chains and 2 beta globin chain. In the sickle cell anemia patients, we have a modified hemoglobin, and that hemoglobin appears in a sickle shape. It takes a shape of sickle. RBC, erythrocyte cell, will changes into sickle shape. That is what it is unable to carry oxygen. So here it results into that change in single base pair, which changes the glutamate into valine. This condition results into a diseased condition, and that diseased condition is called as sickle cell anemia. So here, effect of point mutation that inserts, which inserts or delete, insert or deletes a base in structural gene can be better understood by following simple example, and that is called as sickle cell anemia. So here in the sickle cell anemic condition, we can find that this is a DNA, right? Here in the DNA, we have two strands. This is coding strand. This will be your coding strand. And the other strand is template strand, right? Here we can find normal gene as well as sickle cell hemoglobin, that is HbS gene. What happens in the normal gene? We have coding strand with GAG. We have GAG, and by that we'll get CT in the complementary, CTC. So this strand is going to act as a template strand for the mRNA which is transcribed. So what we get again, G, here it is normal, it is not replaced. So complementary A, and here is GAG. So GAG codes for glutamate. So this is a normal sequence of amino acid that is present in the globulin, that is globulin chain of hemoglobin structure. Now, coming to sickled condition, sickle cell anemic condition, where here is a replacement of, here it has to be thiamine, but it is adenine here. So now this adenine will transcribe on the mRNA as CAC. It has to be CTC, but it is CAC in the sickle condition. So it transcribes, transcription happens where it will produce G, T has to come, but this is RNA, U, G. The code has to be GAG. But as there is a replacement of thymine with adenine, it is going to produce a codon, and that is called as GUG. GUG. Now this GUG code for valine in the place of glutamate at the 6th position. Remaining 5 series of the amino acid is similar. There is no change. Valine, histidine, leucine, threonine, and that means tyrosine and proline. Up to here, up to proline, it is similar. But after proline at the 6th position, it has to be glutamate, but instead of glutamate, we have valine here. So at the 6th position, instead of glutamate, we have valine at the 6th position. This appears in the protein structure, but actual change in the single base happens at the DNA, where thymine, in the place of thymine, we have adenine. So here, pyrimidine is replaced with a purine. That is what this is called as this mutation is called as a transversion mutation. So here, effect of point mutation, that inserts or delete. Here what happened? It is replaced, right? Now, so this is all happened because of a single base pair change. Single base pair change happens at the DNA, which is carried forwarded to the mRNA, and therefore there is a change in the code which leads to the change in the amino acid that is appearing in the sequence of the amino acids in the protein. So this is how there is a replacement of glutamate with valine happens just because of a change in single base pair. That is, in the place of thymine, we have adenine in the DNA, and that is carried forwarded to the mRNA, and from mRNA to the protein. Now, so let's understand about additions and deletions. This is all about single base pair change. Now, come here. So, in order to understand the effect of insertions or deletions, consider a statement, just a statement where it is taken as an example. Any sentence for that matter, you can take it as an example, which is having a 3-letter word containing sentence. That's it. So, consider a statement that is made up of following words, each having 3 letters. Why we are very specific to select the words containing 3 letters? Because codon is a triplet. So, genetic code, each codon is a triplet. That is what we have selected any sentences to that matter. For example, Tom ate bun. Any example you can take. Run was fun. Similarly, the example which were given in your NCERT textbook, here is Ram. It's a 3-letter word, right? Has red cap. In this example, it is just to compare with the codon, and each and every codon is a triplet. So here, each word is compared with a single codon. That's it. Now, in order to understand the effect of insertion or effect of deletion, let's understand. So here, we first understand insertions. For that, we keep on inserting one letter after one letter and see the rearrangement of the statement, how it will look like, right? Now, so first thing, we'll insert B. Here, we'll insert B. Let's see. Ram has, after has and before red, we insert letter B. Take another color. Okay, B, right? So, the next letter R, E. Because it is a triplet, we are giving this D letter to another code, which goes like D, C, A. Let this letter be free. Now, insert another letter, I. Same, Ram has, along with B, Now we are inserting another letter, that is I. Now we'll have only R, continuation of R in this present codon. Then E will shift to another codon, that is D, C. E, D, C. A will shift to another codon. Similarly, here we have inserted BI, no? Now, let's insert BIG. So, Ram has insertion of one complete codon, BIC. Okay. Then, this R will shift to next codon and this C will shift to next codon. So, here what happened is, in this statement, this is a virginal statement, right? We keep on inserting one letter after one letter. So if we insert one base, there is an alteration in the remaining from the moment of the insertion, the remaining codons will all change. And if we insert two letters, the same thing happens from the point of the insertion, the codons keeps on changing. That is, alteration in the reading of the frame is observed. If you insert 3, as a codon is a triplet, once you insert 3 letters or multiples of 3, then what happens? There is no alteration seen after the point of insertion. Say this is complete codon. After that, red cap, which is similar to the virginal statement. So here there is a change up to when there is an insertion of one letter or two letter. If we insert 3 letters, as the 3 letter is a triplet, codon, complete codon has been inserted. So there is no alteration can be seen from that point of attachment, that is insertion. After that, it will be similar to the virginal statement. Now, when we insert it, just the thing what we have, the exercise what we did, the same thing here. If we insert a letter B, this is what happened here, right? In between has and read and rearrange the statement. Rearrange the statement, it would read like this. Here the thing is, as there is an insertion of the one letter, the remaining all codons keeps on changing. Simple. The same thing happened. If you insert 2, again there is an alteration in the remaining codons also. If now we insert 3 letters, as the 3 letter is a triplet, then there is an insertion of one amino acid. And the rest of the codons will be unaltered, will be same as the virginal statement, right? Now, same thing, this is all about insertion. Let's talk about deletion. Similarly, the same exercise can be repeated by deleting letters R, E and D one by one and rearrange the statement to make a triplet word. So here, this is a virginal statement where we have been deleting first R. If we delete R, Ram has, we deleted R, right? And the next letter will come from the next word, C. So, A, P. We'll go for the next codon. Similarly, Ram has, this time along with the R, E also deleted out. D, C, A. Next, P letter will go for the next codon. Now, Ram has, along with R, E, now D is also deleted. C, A, P, which is the same codon as that of the virginal statement, right? So, the conclusion from the above exercise is very obvious. Insertion or deletion of one or two bases changes the reading frame. Until it is 3-letter, the change in the entire reading frame is noticed. So the change in the entire reading frame from the point of insertion or the point of deletion. Now, however, such mutations are referred to as frame shift mutations happened by the insertion or a deletion. That is what we call it as a frame shift insertions or frame shift deletions. So, if there is an insertion or the deletion of one or two bases, the entire frame is going to be shift. And that shift, what we call it as a frame shift insertion mutations or frame shift deletion mutations. Now, so here you can see that comparing original statement with the insertion as well as deletion. If a triplet, if a code completely, that is if a word containing 3 letters completely inserted or deleted, then that is where we don't find any frame shifts insertions or frame shifts deletions, because the entire codon has been inserted or the entire codon has been deleted. So here where, if you consider this as a codon, complete codon, right, 3-letter containing word, it is inserted. So there is no shift in the further codons. Same way, red is deleted. So there is no alteration happen, change happen in the next codon. So until and unless there is an insertion of complete codon, we can find this frame shift in insertions or the frame shift deletions. That is the point. Simple. So here insertion or deletion of 3, if it is coming 3, then it is going to unalter the remaining sequence. That is the unaltered remaining codons. That's it. So insertion or deletion of 3 or multiples of 3 or its multiple bases, insert or delete in one or multiple codon, hence one or multiple amino acids. And reading frame remains unaltered. Reading frame remains unaltered from that point of insertion or deletion. That is what? From that point onwards. Here, one amino acid completely miss in the deletion, whereas an additional amino acid will come. But the remaining sequence of the amino acids will be unaltered. That is the thing. Now, so here, moving on to understand the structure of the tRNA, very, very important to understand the process of the translation, that is the production of the proteins by using the bases which are present on the mRNA. Now, tRNA, transfer RNA molecule, which is going to act as an adapter. So here, we know that amino acids will be present, and amino acids don't have any special speciality to identify what is code that are present on the mRNA. So that work is assigned to the tRNA. This tRNA will have the arms, where one arm we find anticodon, which is a complementary to the codon sequence which is present on the mRNA. By that, it will identify amino acid, and after identifying, it will bind to that amino acid using another arm in its structure. So it will get that the tRNA binds to the amino acid, and this tRNA combining with the amino acid will come to the location where mRNA, that is protein synthesis, is happening. Now, from the very beginning of the proposition of code, that is genetic code, it was clear that Francis Crick, that there has to be a mechanism, to read the code, to read the genetic code, and also to link it to the amino acids. See, if you consider this is a mRNA, we have codons on it. And smaller subunit of the ribosome and larger subunit of the ribosomes attaches to it. This mRNA is attached to the smaller subunit of the ribosome. Then it will get attached to the larger subunit of the ribosome. Now, while this is the movement is happening, according to the codons which are present on the mRNA, the amino acids which are present in the pool has to be brought to the site. and that work is done by the tRNA. That is what we call it as a transfer RNA. tRNA, transfer RNA. Now, so because amino acid has no structural specialities to read the code uniquely. The code is present on the mRNA. Amino acids are present in the pool. According to the code which is present on the mRNA, amino acids has to be identified and it has to be brought to the site where the protein synthesis is happening. And that work is performed by the tRNA. Now, if you look at tRNA structure, so here is the arm which binds to amino acid, and that is what we call it as an acceptor arm. Opposite to that, we have anticodon arm. We have variable arm. We have T size pseudouridine, thymine, pseudouridine, and cytosine containing arm. Similarly, dihydrouridine arm. So, if you look at the structure of the tRNA have hands, arms. One is to have the anticodon, which can complementary, that is it is showing the complementarity base pair with the codons which are present on the mRNA. So that is what it can read because it is having anticodons on it. And opposite to that, by reading the codons, by using the anticodons, it can identify a specific amino acid, and that amino acid will be bind to this arm, and this arm is called as acceptor arm. So this is all about the structure of tRNA. So Crick postulated the presence of adapter molecule, and that adapter molecule is nothing but tRNA. That would on one hand can read the code because it is having an anticodon arm, and another hand would bind to a specific amino acid, and the specific amino acids can be identified by reading the codons by using the anticodon arm, because complementarity base pair exists between the codons and anticodons. So look at here, this is an mRNA which is having codons on it, right? And to this codon there is an anticodon. With this, it can identify to which amino acid it has to bind. To which amino acid this tRNA is going to be bind is decided by the codon which is present on the mRNA, right? So, and this amino acid will come to the site of the protein synthesis. So here, the tRNA, then previously it was called as a soluble RNA, was known much before the genetic code was postulated. However, its role as an adapter, where it is going to identify the amino acid, specific amino acids, and it is going to bind to that amino acid and get to the site of the protein synthesis. That adapter molecule function was assigned to the tRNAs much later. Now, but it is very well known as a soluble RNA. Once its work has been assigned as an adapter molecule, then it got the name like tRNA, from soluble RNA to tRNA, that is transfer RNA. Why we have given the name transfer RNA? Because it is transferring the amino acids from the pool to the site where the protein synthesis is happening. tRNA has an anticodon loop. Just now I said no, anticodon loop, which is useful to show the complementarity towards the codons which are present on the mRNA. And based on the codon which is present on the mRNA, by attaching with this anticodon, it is going to identify a specific amino acid, and that specific amino acid is going to bind to an other hand, that is other arm, other loop of the tRNA. See, bases complementary to the code. Complementary to the code. It is having AGU, and the complementary code will be UCA. So, AGU will code for serine. And it also has an amino acid acceptor end. On the other hand, it has an acceptor arm which can accept a specific amino acid, and that specific amino acid is based upon the code which is present on the mRNA. This is important. So, whatever the information that is given to the tRNA is coming from the mRNA, and we already know that the mRNA will get the information from ultimately from DNA. That is what the genes are finally first transcribed, then translated into protein. The structure of the protein is decided by the sequence of the DNA which is present in the gene. Now, it also has an amino acid acceptor end to which it binds to the amino acid. By this, it is very clear. So, here, and initiation. See, while we are discussing during the transcription, we come across the initiation, elongation and termination. Similarly, these steps will be followed in the translation also, like initiation of the translation, elongation of the translation and termination of the translation. So, for initiation of translation, there is an other specific tRNA, and it is refers to as initiator RNA. Just like in codons, we have got this stop codon and start codon, right? Similarly, here also we have an initiation special tRNA, initiator tRNA. Now, coming to the stop codons, while we are discussing about the genetic code, totally 64 codons are there, out of which 3 will act as the stop codons and 1 will initiate the process of the protein synthesis, what we call it as a start codon or initiator codon, that is called as AUG, which code for the methionine. This is all the things we already discussed. They are no RNA, that means tRNA for stop codons, because it is resulting in no amino acids, because it is not going to code for any amino acids. That is what, there is no specific tRNAs that are available for the stop codons, right? Now, the secondary structure of the tRNA has been disputed, which looks like a clover leaf. This is what? This is the clover leaf model, secondary structure of the tRNA. In actual structure, the tRNA is a compact molecule which will look like an inverted alphabet L. See, usually mRNA is just like a thread, right? It is just like a thread. I hope this is visible to you. Then, this is mRNA, right? Even every RNA will be like a single stranded thread-like structure. It will fold in such a way. This is all just folding. You can find 5' and 3'. So, actually, if you take it off, it will come like a single thread. This single thread will fold in such a way, it is going to look like a clover leaf model. That's it, right? Yeah, this is all about the structure of the tRNA and moreover genetic code and mutations. In this class, we came across mutations, different types of the mutations like point mutation, frame shift mutation. Frame shift mutations happens with 1 or 2 insertions or deletions. If the further frame shift, if it is unaltered, that can be seen by the insertion of the multiples of 3 or 3 letters, that is 3 codon. Now, with this, we come to the end of mutations and genetic code, as well as we discussed about the structure of the tRNA. In the coming up session, let's come up with a next important process, and that is called as translation, protein synthesis. Until then, keep learning with vLearning. Thank you.